The interface has four main elements: the gene expression matrix, the sample view, the gene view, the bottom side view, that shows the expressions of all the genes for a sample, and the left side view, that shows the expressions of all the samples for a gene. At every moment, the current column and row of the mouse select a gene and a sample. The current sample and gene are shown with a cross in the sample and the gene view. The user can zoom in to get details in the gem, and also see textual information for the current selection of sample and gene. Zoom can also be used in the sample and the gene view to view details. Box selections in the gem reveal the selected samples and genes in the sample and gene view. In the gem, 3 clusters can be easily located. The first cluster contains the pheochromocytoma and paraganglioma. The second corresponds to kidney clear cell renal cancer. Finally, the bottom one corresponds to kidney normal tissue. A small subcluster inside the first one, with different genetic behavior, appears here. Zooming in the sample view, the researcher can identify the samples, which in this case correspond to tumors with mutations in the VHL gene. This other subcluster contains samples from both tumors that overexpress in the same way a set of genes. Zooming in the gene view, the researcher can check that it mostly corresponds to genes involved in angiogenesis and extracellular matrix development. Alternatively, the gem can be reorganized using standard B-clustering algorithms. The interaction is the same as we've seen before. The user can also do precise selections in the sample and gene view that are lively shown in the gem. The researcher can also discover genes with similar behavior for a subgroup of samples. In this case, the user selects samples of kidney renal clear cell carcinoma and VHL mutated pheochromocytomas, looking for a set of genes that behave similarly in both conditions. Then, he can rearrange the gene view so that genes with similar behavior for both conditions are closely located. The gene SLC16A3 is essential for cancer cell survival and predicts tumor progression in patients with clear cell renal cancer, and has also been biochemically associated with VHL-regulated signaling pathways. Therefore, it is expected to be upregulated in VHL-deficient tumors. A simple inspection reveals how some of the neighbor genes, such as ALDO or ENO1, behave in a similar manner. Additionally, the user can explore similarities and differences between tumors with specific gene mutations. For instance, we can explore the behavior with respect to genes involved in angiogenesis. The user can introduce the corresponding genes and rearrange the sample view to discover similarities among the samples. We see that the normal tissues group apart. Clear cell renal cancer is grouped in this area, and pheochromocytoma does in this area. It is interesting to locate the pheochromocytomas carrying VHL mutations. It can be seen that they group together near renal clear cell carcinomas, which means that they behave similarly according to angiogenesis. However, doing the same for the genes involved in glycolysis, the 3 types of tissues are clearly separated, which means they have different behaviors according to glycolysis. But remarkably, tumors carrying VHL mutations group with kidney tumors, revealing that activation of glycolysis is a feature of this kind of tumors. Down here, group normal tissues. In this area kidney tumors, and here the pheochromocytomas.